just an idea where someone can say, oh, I'd like to know more about these species and how they interact. So let's see. Okay. So go ahead, please, Danny.
Yeah.
Okay. So there are a couple of concepts I have to talk to you about. One of them is invasive species, what they mean, how do you define them? Is darlingite really an invasive species? And the other is, if they're not invasive, are they displacing another species? And if they're doing that, how do we know? How do you prove that it's happening? And what are the ecological mechanisms that force this to occur? Okay. Next. So this is what I'm going to be talking about briefly, and I like outlines because they force me to more or less stick to the topic. Otherwise, I just kind of go off, a little bit into the weeds. So I'll talk to you a little bit about things you might even know about, the ecology and vector biology of darling, because it's the most studied malaria species in all of Latin America, because it's so important for the Amazon basin. I'm going to talk to you a little bit about what is environmental niche modeling for someone who don't know. It's not so complicated, but you need to have a general idea of how to do it, what it means. And we're going to talk about the characteristics of an actual invasive species, and then about darlingite and thinking, why do people call darlingite invasive if it's not formally an invasive species? And I'll give you a couple of really great examples of invasive species, so you can see and you can think about darlingite in that context. I'm going to make sure you have a sense of what's the difference between invasion and displacement, and how do you recognize if displacement is ongoing now, or is it happening in the past? Not more frequent, but you could start to get a sense if you have historical records. And then how would you collect data? What kinds of data do you need to demonstrate that this is happening? You can't always know if it's occurring. Sometimes you just collect mosquitoes to try to find out if there are trends. It's very important to keep records, to collect in sometimes very odd places. If you're focused on, we're often focused on health, but we need to be also focused on biodiversity, even the mosquitoes, because they're very important to many other organisms that are not there by themselves in isolation. And is there a case to be made for darlingite displacing Benarope in some places, or no places? We don't have the evidence to know, but we have some very interesting data from 2003 that could be used to test this hypothesis. Okay, next. So the bioecology, you probably have a pretty good idea, but I'm going to remind you that not everywhere where darlingite is does rainfall dictate to such an extent the rise and fall of the population across the year. But here in Amazonian Peru, it's essential because rainfall creates new breeding sites. The water level rises 10 meters during the rainy season. It's a huge amount. And this creates many breeding sites for darlingite, but also for other species. So it's very important. Rainfall itself, of course, not just the melt from the Andes that's coming down in the mountains, but also where can the mosquitoes find a blood meal? Are there lots of chickens? Are there lots of humans? Are there any cattle? What else could they feed on? They're very broad. They're quite generalist, actually. And where do they breed? They're actually becoming almost generalist breeders. They prefer some things, but they're pretty broad. It's very interesting. Usually clear, deep water with low light, but not always and not only. The adults like the ecotone or the forest fringe. So when you'll see further along when people are logging forest and doing various deforestation for mining, for agricultural settlements, for dam construction, for many reasons, for highway construction, they often create leave behind blocks, forest fragment blocks. It turns out that all around those edges, which are very long, if you think about linear, if you go out and measure the edges, lots and lots and lots of meters, is just where darling likes to be. So it's perfect setup for them. They like a low vegetation index. What does that mean? It means they aren't usually found in big shady forests. They're found where there's a break in the forest, where there's an opening in the forest, much more so than actually deep in the forest. They can be found in fish ponds, in lots of places where there's mining, especially gold mining, because of the way it's constructed, because it first of all requires a lot of deforestation and then creation of these really pretty ugly ponds and pools. But those places become, as soon as the mercury is not quite so toxic, they become nice places for darling and other distinguished breeding. Flooded forests, streams, ponds, so they're very widespread. Next. So here, environmental niche modeling is not so complicated. It predicts based on distributions that are already extant, that exist in the literature that people can find. They have to have the proper latitude and longitude. They have to be verified by somebody who knows enough about GIS systems to do that. But the models are with the predictor variables that are the abiotic things, the climate variables, the vegetation, the land use, and they're based on the distributions that people have created over years for species, different species in their region or in their local area or in the whole country, depending. What's wrong with this? It's not that it's wrong, it's simply very limited. And why is it very limited? Because it's not local. It uses sort of a broad sweep of where the species should be based on where it's been in the past. The distributions are only correlative. They don't tell us why they might be there or what they're really prefer, apart from these the climate variables, which is only one aspect of what allows a mosquito or any other species to be found to move around, to redistribute itself, to migrate, to move. And the other problem with these abiotic characteristics is they're changing all the time because Earth keeps on warming. So the models that are created have to be constantly upgraded and updated because they're simply not accurate. They're accurate for a very limited time and then need to be continually monitored, which is actually great. It requires people to keep on working on them and not let them become static. They should be considered a living, evolving thing. But they don't consider interactions with other species, and that's a huge piece of what keeps species alive. They interact with predators, competitors, and mutualists, and then those should be incorporated into predictions. They're much more difficult data to get, which is why people are not doing this. But if you really want to understand whether species are displacing each other or invading an area and why, you have to understand these other pieces. There's a very nice paper by Steve Philonubis and Steve Giuliano that talks about this. They're very both. They're both very good ecologists, and they're focused on this quite nicely. But we need to we could so easily get a distribution map in for darling eye. And it would be so nice to have one, because the one that exists, to go back a couple, is Cinca from 2012. So that actually the data were from before 2012, because that's when it was published. Peru doesn't have darling eye where all the places where it actually has darling. This is on a big scale. So they are missing a whole bunch of data. And besides the 2012, there's lots of new data on darling distribution since then. Okay, let's keep going. Actually, 2010, that paper. What it is an invasive species? You all sort of have an idea, but you might not know specifics. It's they're carried passively. People don't deliberately bring invasive species into another continent unless they're, conquistadores or explorers or people who are coming to the new world, thinking that they would bring the old world with them, bring all their plants, bring all their animals with them, which is in most cases a complete disaster. Because those imported plants have often outcompeted the natives and destroy them or replace them, which is not very great. But the biggest concerns are the negative effect on endemic biodiversity, and also the establishment of species that can transmit, not just we worry about the humans, but there's so many animal viruses as well, and plant pathogens that destroy native crops or destroy native forests. They're just as important. What do you define as a successful invasive species? It has to produce enough population, in other words, enough offspring, that population has to be able to grow. So if you're thinking theoretically, you'd be seeing a curve going up over time, obviously. So they need to have enough to initiate, but then maintain the population, and then they have to have enough to spread out and keep on searching for more habitat that they like, the appropriate climate, the appropriate latitude, rainfall, etc. If the initial invasion is successful and they're really making great use of that new habitat, they're likely to be able to spread further. They're often introduced. People sometimes are really actually thinking about whether they should put introduce. They'll look at the latitude, the temperature of the climate, and say, okay, this is thriving over here in Australia, it's definitely going to thrive here. And let's say if they were going from New Zealand to Chile, for instance, those are almost parallel. And there are some species that the continents together. So there's still some species that live in both places. That's not unusual. But this is different. This is a much more complex. It's not just relic populations that used to be together in this huge supercontinent. But the other thing to remember is once you've introduced something, it just keeps changing over time. It never remains static. None of these things are static. They're all changing. Their distributions keep moving, which is fascinating, but it's more challenging to keep up with them next. Here's a great example. The best one maybe is Aedes albopicus that was introduced into Texas in 1987. Many of you will be aware. Hi, hola, bienvenida. Many of you will be aware that it's invaded all kinds of parts of Latin America and not, so it's not just Texas. Texas is one of the first places it came and was very successful from Southeast Asia. And it turns out in Southeast Asia, the conditions between that area of Southeast Asia and Texas are very similar, so it's done extremely well. But it's also been spread all over the world by used tires. And humans are so stupid that we think that spreading around used tires and shipping them from one country to another is a really good idea. The reason it's not very smart is because these Aedes albopicus and Aegypti, which is just as bad, their eggs can stay dormant and dried out for more than a year. And then you just have to put them in water like a little toy when you were a kid. You put a sponge thing in water and like, it's just, they do very well. They'll just come to life. If you didn't know that the ecology was that they were actually eggs, you would think it's magic. Here come the larvae. So what, in this case of Aedes albopicus, what really amplified its invasion, there's a lot of things. They're continual breeders. They are almost always in enclosed spaces, but there's so many things they can lay eggs in a bottle catch of water on the ground. If you start thinking you want to control them by removing all these breeding sites, you'd have to persuade every single person not to keep water in any container outside their or inside their houses, or have a screen over top. It's not very practical. It's a nice idea to try to educate everybody, but it's not going to be equally efficient for everybody. They're opportunistic. They really will prefer to feed on humans. They're day bikers. So in fact, you're very vulnerable when you're sitting anywhere outside where there's a lot of Aedes, because they'll, you're not going to get a mosquito net in the middle of the day. You're going to be sitting around somewhere eating or whatever you're doing. They're going to get, come and bite you behind your knees or something. They're very tricky biters, but they do prefer humans. They have this diapause. Where Aedes aegypti is, they co-occur with them. Sometimes one will outcompete the other, but they're very interesting because it's almost on a case-by-case basis because it depends on the climate and the local temperature, the habitat, the humidity, everything about it, even the containers. So it's hard to predict who's going to displace who. In some places, Aedes aegypti, for instance, in southern Florida and Bermuda and several other places, the two of them co-occur. You can't always tell which one they're going to replace the other. Albopictus presence in Europe, so now it's all over Europe too, but it's driven mostly by rainfall and daytime surface temperature. That was in 2013. So what that saying is now saying is that they're continuing to expand northward. So they're not going to stop just because you would like them not to keep going. They're going to keep spreading next. Another, this is very disturbing. You don't know anything about not Stevens eye. It's I think it first showed up about 2015, maybe in the Horn of Africa. It's a very good Asian vector, extremely efficient, and unfortunately, it's now in several parts of Africa. And it's both, it's very good at transmitting both toxicity and vivax, and it's urban. So the reality in Africa is most of the vector interventions that people have historically been doing and still are doing, they are for rural mosquitoes. They are not prepared not prepared to manage yet container breeding mosquitoes in the urban centers that are anopheles that are transmittable area. It's considered a rural disease, and that's where the focus almost always is. They're going to have to now spread their very thin resources in several of these countries and try and focus both on the rural areas where Gambier, Fenestis, Clucia, and Gambier, Arabiensis, where those are the main vectors. In addition, in the urban centers where Stevens eye is now really becoming a serious problem. There's a whole other story to this that vivax, people often say, well, there's not any vivax in Africa, but there's more and more and more of it. So it's also a whole other very fascinating story for parasitologists. These are very straightforward. It may not sound like they're straightforward, but they're clearly coming from a different continent. They're clearly coming from another area and displacing native species. Each of these mosquitoes came without their predators, parasites, and normal competitors, which is one of the reasons that they're doing very well with this spread very quickly. Next. So what about darling eye? What things do we already know about darling eye? We found out in the past 20 years in the literature, people have done lots of work on darling eye. We know that they really colonize rapidly these modified landscapes. Before that we've known about this since Charles' early work in 1996 in Brazil, which was wonderful. But if you look at forests, Amy Victor did a fabulous study in 2009 all along the Iquitos Canal highway, where people had started building fish ponds. And she did a very nice study of transects all over the place, looking at different layers of forest, different percentages of forest cover, I should say. And she found almost none in the deep forest, but lots in the secondary forest or where there've been recent deforestation. The correlation is really superb. It reduces the diversity of other anomalous species. Now, we don't have absolute evidence that that's for darling. We don't really know. We think we have anecdotal information, but the Ministry of Health, I believe, has lots and lots of files that can be accessed, and those can be digitized and they can be analyzed because they have some very good records of species distribution, but they're not available. But what is it that's prompting darling eye to invade these areas? What is it about deforestation? Does it put opens up some sunlight? Does it open up ponds? What is it? Why are they? Partly there's humans there, but there's a lot of possible reasons. The low NDVI, which already discussed briefly, the size or the frequency of the equatorial areas could be, you'll see in a little bit in Brazil how many of them there are. Patch sizes, the characteristics of the breeding sites themselves, and lots of humans, and it's a very available blood meal because humans, we're not very good at getting under bed nets at sundown because we're not babies. You can put a baby under a net, but you can't put us under nets, us adults. We don't want to be under a net at 6 at night. We like to do other things. We like to eat outside. We like to go and dance. We like to drink beer. We like to do lots of things, go swimming in the river, whatever it is, we're not going to be carrying around our insect, our mosquito nets with us everywhere. So we're very vulnerable. Okay, next. So the difference between an invasive species and the displacement of one species to the other is that when two invasive species occupy a very similar ecological niche, one usually displaces the other because they really are not very good at sharing. We can say, like you say about some people, they don't play well with others. It's exactly the same with these mosquitoes. This leads to what's called habitat segregation. One species is particularly good at surviving right here, and the other one goes over there. Or they sort of go back and forth until they get some kind of equilibrium. Or as you saw, or as you didn't see, if you didn't read the paper, but I read them, the distribution of Aedes and albopictus in these containers and retrievals changes over time. So it doesn't stay static, right? But they do move around and displace each other. Actually, it's very interesting. There's another interspecific mating bias, which what it means is that the males of albopictus mate with the females of Aedes aegypti, and the females don't have very many offspring. But when yes, those females have offspring, but if the male Aedes aegypti mates the female albopictus, no, I have that. Never mind. You get the idea. Albopictus is more successful in this mating interference than Aedes aegypti is. So there'll be more eggs of albopictus in each batch and fewer of Aedes aegypti. So eventually they move over. But what happens? These are both invasive species. What happens if you have two species that are native, like Benaroki and Darling? What happens in that case? We don't actually have a mechanism for that because nobody's bothered to study it. So if the displacer, in this case, in our case, Darlingite, is more effective at transmitting malaria than a species that's displacing, we should care about it more because we're going to have better, more efficient malaria vectors only in lots of places if they keep on displacing and replacing the vectors or the species that are in a similar niche, but they're not as good at transmitting malaria. So we need some data about if you want to study whether Darlingite is actually displaced Benaroki in Peru, we need to get some data next. The simplest terms are what I was talking about trying to get hold of more data from the Ministry of Health to see if we could actually demonstrate that there's more Darlingite in all of these different localities and less Benaroki or less of other forest species than there used to be over time. You could start 20 years ago. There's a paper by George Shaler et al. In 2003. That would be a great place to start. I'll show you the map in 2 minutes. The harder thing to do is not that challenging to collect the adult mosquitoes. You sit and you're the human beta. It's just not overwhelmingly complicated to do that. You have to be safe, but it's not a rocket science. But getting to the aquatic, getting to the breeding sites is much more challenging. It takes more time. You have to map them, you have to find them, you have to figure out which ones are productive. It's a lot more work, but it's where the interaction is more likely to happen. We're not in a competitive environment for the adults where they're competing for space on human skin to take blood from. That's not what's happening. It's happening in the aquatic stage, and we don't know enough about exactly how, where the larvae are all living at exactly the same height in the water column. Are they feeding on exactly the same microorganisms, etc.? We don't have that information. But the traits of invasive species, or those with what we call displacement potential, like Darlingite, are that they occupy human-dominated habitats. They're associated strongly with disturbance like deforestation and land building and mining. They have a high maximum rate of population increase we don't have very good data on that, but when darling eye invaded the Iquitos in the 90s, in the late 90s, the abundance of darling eye and then the abundance of thousands and thousands of malaria cases is very good evidence for that, although people haven't necessarily been bothering to quantify it. It could be done. The expanding human population in settlements, if people keep expanding and growing and moving into the forest, of course, there's going to be more suitable and underexploited microhabitats that darling eye and other species like it that are nicely adapted to this kind of deforested area are going to thrive in. Next. So if we're going to look for evidence, what kind of data do we need? We need to know more about the actual ecological niches of darling eye and Veneroki. We know a lot about darling eye, but we know very little about Veneroki. The sort of routine way of doing this is collecting monthly, which is really good. There's a very nice paper by Bennett et al. 2021, where this was exactly done in Panama. They did it monthly for 2 years, and I have very nice data showing the evidence that they can show that they've got, I think it's Aedes albopictus, busy displacing Aedes aegypti in some areas, but not all of the areas. So, and that's the key. That's what's so interesting about it. 2 years is just, it happens that it's useful to have 24 data points tenfold. If the scope is local, you want to choose localities that are relatively similar because both the mosquito distribution and the humans and parasites are notoriously heterogeneous. So if you want to at least test that there's been a change, you need to kind of start with the habitats that are as similar as possible. And then you can look more broadly once you have that in place. As many records as possible. And I admit that the aquatic stages are hard to find for the records. The adult stages are not so hard to find. So that would be a great starting point next. So this is a very nice piece of work. It was really underappreciated in my personal opinion. These folks in 2000 collected in all of those places. They found darling eye in many of them, but they found Veneroki in 71% of these localities, enormously broadly distributed throughout the Amazon, really remarkable. They had several, probably at least 100 localities. They just did an amazing job, and they did not find darling eye in some places, but for example, these are other circles. For example, it would be wonderful to go into these areas and see if there's darling eye now, or here, if there's darling eye here now, or here. There's lots of possibilities just from looking at this particular map. Anyway, that's what I would do if I were queen for the day, and I were in charge of these collections and through. That's where I would start. Next. And then we have some data from my research technician and I, Sarah, scoured the literature and looked for newer data just to see where darling eye and Veneroki are distributed. So if we could do it in preparation for this talk, somebody who was seriously looking to get additional records from the Ministry of Health and maybe some of the Spanish literature which we didn't see in detail. This is not always very easy to find it. And look and see where are they exactly co-occurring. Because if you don't have the GPS data and somebody just said lacuna, you don't know if you actually look like in the same stream where they're seeing the houses, but that's kind of important. Okay, next. So this is the aquatic. Well, we've done some larval work, but not that much. It's never been our mandate, but one of my grad students, Kate Pressing, we decided to look at for other reasons, but we found Veneroki bee and darling eye together in water bodies in 5 localities: Lacuna and Nueva Horizonte and Triunfoso along the Nauta Highway, but also Lacuna's on the Nanay River. We also found them in SEM, which is much further away, and also way up here in Libertad. And that's quite a distribution just in the Quito's area. So we know Veneroki's around, but it's not nearly as common as darling eye is. But we weren't looking for Veneroki. We were just looking for and identifying what we found. So if we were more focused on Benroki, we might have a better success next. So the ecological niche, we basically talked about it, but it does need some kind of forest opening or ecotone. It needs roads and tracks and rainwater cooling along the roadside, probably. When people are building roads with deforestation, non-permanent anthropogenic change in the natural environment with low NEI, lowlands, moderately hilly. And then the breeding sites, we've talked about all of these already. They really like edge vegetation. If you're in a collective darling eye in a pond, you're going to find them near the edges. Why? Mostly because that's where the low water vegetation is, and they can be hiding from predators, aquatic predators. They're in among the reeds or in among the algae on the surface. Both of those are very useful for them. Next. Benroki, we don't know anything at all because the old records of the larvae were Benroki sexulatu, and there might be 2 species of Benroki. Maybe they're all B, and we don't actually know. Yeah. Next. If you're thinking about how come darling eye is so good at adapting and moving into these environments, it's because they have something called phenotypic plasticity, which lots of organisms have. But darling eye has it in spades. It's the ability of individual genotypes to produce different or novel phenotypes, which is phenotype is what you see in terms of external characteristics as opposed to the genetics that are driving the genes themselves in a changing environment. In other words, they're programmed to be adaptable. If any of you remembered or looked at the genomes of Anopheles gambiae, the most striking thing was the amount of variation in the whole genome is staggering. So they're able to modify and change just about in any situation. That's an amazing quality to have, and darling has it too. There's lots of evidence for genetic differentiation. There's lots of evidence for very few barriers to actual gene flow. There's surprisingly low level of population differentiation because they're actually flying and they're moving around in the rivers probably. There's lots of variable behavior. We already know that they feed indoors and outdoors. They feed on humans, they feed on birds. They rest outdoors, but they're very opportunistic, as a matter of fact. They try to keep people away from them, they'll feed on whatever else is around them. So we did a couple of very nice life history lab studies on darling, and demonstrated that they have tremendous plasticity, which is in the next slide. Don't worry about the details. What I'm showing you is that if you rear these individual families from all of these different places, some at the same latitude, these guys, and then some at very different latitudes, but the same longitude here. You can see that in every case, the comparisons show that the temperature is significantly altered all the traits. You have faster larval development, shorter adult life and overall lifespan, and smaller body sizes as the temperatures got colder. And the genetic variation and plasticity among the families from single localities, but also in the region, in the different regions. The second study that was in process and vectors. So this is the raw material for natural selection. Next. This is Leo Chavez's work, our wonderful postdoc in Brazil, who worked with in this other project. And he showed that looked over, let's see, 7 years in huge numbers of municipalities all across the Amazon, and to look at deforestation rate, the degraded and impacted forest, and correlate that with malaria cases, which show that 1 kilometer square of deforestation produces 27 million malaria cases. A lot of malaria cases. There was a huge positive correlation between the number of impacted forest patches that were less than 5 kilometers square. That's not very big. That's the standard size in terms of, I think 95% of all the patch sizes in this huge study were that size or smaller. So it seems to have become a sort of odd standard that the patch size will be this big. It's probably mandated by the environmental folks at the Obama initially to say, you have to leave a stream, you have to leave a water course. If you have a row of trees along the stream, you can't cut them down. So all of these places all around their edges are wonderful places to be. So interesting. They're probably, these patch sizes are also linked to the partial shade of the larval habitat that Darwin likes. So, okay, next. Okay, so it would be really useful, I think, to look. Somebody decided that this could be their project for a master's or a PhD, probably a PhD, or even a postdoc. That would be really cool. Would be really useful to understand how Darling Eye is getting around in other places. Darling Eye has lots of characteristics, but we don't know anything about the larval stage really. It's one of the competition in that is very valuable as well. We already know that there will be cold furs in the larval sites for Darling Eye, but sorry, we also know the adult cold fur in many localities in Dayton, Vilna, and Jan province. We saw that in the Shaler et al. Map, but we also re-analyzed some data in 2018. The paper just came out, but we don't know anything about the mechanism. We don't know very much about the ecological conditions, especially as I've now said about 10 times. I'm better of reading. It would be nice to focus on mechanisms that might facilitate invasiveness leading to gene flow and connectivity across these very heterogeneous landscapes. But there's malaria in all of them, which is what a very interesting emerging factor next. I think that's actually all. Okay. It was a little speedy, but I thought I was giving this talk tomorrow, so I finished it in peace early this morning. So there you go. I'm a little wired, but never mind. Get another cup of coffee and keep me going for this talking. Oh, you're all stumped and you're thinking so deeply about displacement and invasion and Darwin and general that you can't think of a simple question. Yes.
Thank you, Jan. It was a very comprehensive talk. So I'm curious about when you talk about displacement, and you mentioned something about mating different species.
Yes.
But are there, what are the other strategies that the mosquitoes use for displacement?
Those are the main, well, the competitive displacement in the larval stage is the main, the two main ones are that. Those are the only two I could find. I read a lot of papers. There's more theory. There's not very much practical information, but those are the two that people have actually demonstrated that the cross mating is very uneven in those two species, which is fascinating because they're almost sister taxa. They're very closely related genetically. So that's fascinating too, but it's one of the reasons they're different species. Right? Of course. So yeah, those are the ones that are considered the most likely to be important in invasion in mosquito species. Now, any other species, I don't know. Yeah.
Yes. So you were saying indeed, you cannot ask the people to be, to protect themselves all day. They use bed nets and so on.
It's challenging.
Yeah.
I'm saying they could use repellents. There are options, but they're not. When you're talking about a day biter, it's just more difficult to, I know you're having another dengue outbreak here, and it's very serious, and people can get really sick. I've had dengue, it's awful, and people can get hemorrhagic dengue, and that's really dangerous. So the problem is figuring out where the babies are breeding in your body. That's one of the reasons why there's so many overtraps, and there's very successful at trying to trap out. Darling, having overtraps in a neighborhood where there's dengue, even before you know that there's dengue coming, and actually checking them regularly and finding out where the larvae are, and then you can treat that water. You can treat it with lots of different things. There's bacillus that people can use. You still can use toxuricaides, even though people have sort of forgotten that mosquito eats a lot of the Aedes larvae. And if you can actually see, especially if you're talking about somebody's home. They're a biologist, for example, living in Lima or living nearby, you could persuade them probably to try toxin pennies, because they will very happily eat them, but then you have to find something else for it to eat. It eats up all your babies, so you have to give it something else or it's gonna die. But the bacillus is very safe. You don't want to put it in the drinking water, but you can put it anywhere else. And it does kill them. And it's a biological organism. It doesn't kill anything else. It's pretty specific.
Yeah, because, for example, last week we were in a community and even I forgot to put my repellent.
Yeah,
And I assume, yeah, the inhabitants there, they
they're not gonna
doing
put
it
it
every
on.
day, it's like,
and you sweat so much sometimes
Yeah, yeah.
you can't keep it on. It's challenging. I'm not saying there are any easy answers. I walk around all over the place, through other places, always in long pants and long shirts, and people are always sort of saying, and I sweat a lot.
Mm.
I'm a lovely person to be around, but I don't get bitten. So, you know, it's really a matter of education, and then people, of course, take their own risks. But you need to have an idea where the cases are, where the cases in a local place. If you're going to travel and you're paying attention, find out where their dengue alerts and try to don't go there, honestly, if you can avoid it. If you're going to try and treat it, that's different. But if you're just a casual visitor, you don't have to go to those places usually.
Yeah.
You know, it's not bad to avoid them. It's not very helpful. I completely agree. But a lot of cities now in Peru have reasonably good efforts in dengue control. They will know where the heck the dengue cases are too, which is also valuable. But if you're just a casual visitor, what's happening? Why were you there?
well, we are about to start a new study, so I wanted to visit the health
Yeah.
post and. Yeah. I put my repellent once after I got the somebody.
Several
Mosquito
bites to the back one.
Yeah.
I mean, that's personal protection, right? I mean, that's
Yeah.
more challenging to manage because people do their own thing or don't. How do you protect people on a bigger scale than personal protection? That's the challenge. Bed nets are simple, relatively speaking. I mean, they have their own challenges. You know, they hold, people don't use them. Okay, that's not very good. But there's nothing quite as effective for day biting mosquitoes. Because we're on a bicycle, so we're not staying in one place. So it's really hard. I don't have a straight solution of it. I mean, except treating the containers are the best ways to treat these container users. That's really is the most intelligent use of resources, is to focus on where the releases are and treat them.
Yeah. I don't think there are studies in also.
Yeah.
Hi. Could the phenotypic plasticity explain the fact that these mosquitoes can be
Yes,
be resistant to plaguicide, insecticides?
yes, but yes, it's genetic actually. It's a genetic mutation. It's
But-
not.
The fact that it's biodiversity, it increases the fact that they are going to be resistant.
Their genetic variation and their ways of responding to things, including to insecticide treatment. Yes, it does, I think, make it easier for them to overcome quite quickly. You're right. It's a good point. It's a good thought. Good way of thinking about it. Yeah. We did a study about 6 months ago, looking at two dominant, the ACE1 gene and the voltage channel gene for insecticide resistance. Those are two of the genes where the mutations show up that are really directly linked to insecticide resistance. And we didn't find any in 500 darling eye that we looked at in several places. And we were a little surprised. But my collaborators at Harvard are doing the whole genome of darling eye, and they found insecticide resistance in French Guiana, where there was a lot of insecticide used agriculturally. And they also have found, and that's in multiple sites, and they've now found one site in Peru, it's not published yet, but it's you'll see it in a post. I think it's Luna, oh no. Coweta, I think it's in Coweta, and that's the only place in Peru so far, but they only have one. They only have mosquitoes that lead into there, they don't have lots of others, and they would like to, but you have to, they need 50 from each place, and it's not simple, it's not just like a little collecting expedition, you have to really think about it. But they have also looked at several places in Brazil where we expected there to be, and there isn't. So one of the complexities of darling eye and mosquitoes in general, where they're very common, like the Amazon basin, is the sheer numbers of the mosquitoes and the amount proportionate you can actually collect. It's not always very representative, and that's really challenging. You have to be very mindful of where you're collecting. It's not a random choice. If you're looking for insecticide resistance, you do have to do what they did in British Guyana, which is go where there's agricultural insecticide use nearby to try and at least see where the signal is. And then you say, okay, this is the signature in the genome of where this should be. Now we can take that information and look much more narrowly at that place in lots of other populations, which is what these guys are in the process. I say that there are two guys doing this in Harvard right now, doing the analysis of many of our samples that we collected. So I don't know what the outcome is. They don't either. They just have the preliminary data from British Guyana in one place, which is pretty exciting. The fact that we didn't see any with our 500, but they were looking at the whole genome is not surprising. The whole genome is so much more informative than 2 genes. It's hard to compare. We didn't have the genome at the time when we started. So.
Otra pregunta. Sí.
Yeah, hi.
We're going right now to the moon.
Yes.
So we have higher amounts of rain and higher temperature. I suppose that's going to change the distribution of the food.
It actually will change all lots of other species, but it certainly will likely have quite an effect on darling eye. The person who knows, I'm not actually an ecologist, and I don't know that much about it, but the person who does is Maria Berlet. She's done some wonderful work in Venezuela on the effects of El Nino, La Nina for 80s. But her work is exemplary and wonderfully useful. So if you're interested in really, she's very approachable. Email her. She works with me very intensively, and she's delightful. So she's really good at helping. So if you get interested in one, think about it.